Hello everyone, welcome to my science channel. I'm Yusun from the Department of Chemistry at UCL, and today I'm going to take you on a fascinating scientific journey exploring how DNA origami may fold the key to a new solution for diabetes. Diabetes is a prevalent and serious metabolic disease characterized by persistent high blood sugar levels. In a healthy state, our body has a sophisticated blood glucose regulation system. First, beta cells in the pancreas secrete insulin. Then, insulin acts on insulin receptors on cell surfaces. Finally, cells absorb glucose from the bloodstream, lowering blood sugar level. However, in pathological conditions, this system can malfunction in two primary ways. For type 1 diabetes, beta cells are damaged and cannot produce sufficient insulin. And for type 2 diabetes, although insulin is produced normally, cells cannot accept it. Regardless of the type, the result is the same. Insulin fails to function properly, leading to chronic high blood sugar levels. If left uncontrolled, diabetes can lead to a series of severe complications. And these complications seriously threaten patients' quality of life. So, are there any treatment methods for diabetes? As we previously discussed, the key point of diabetes treatment is to control blood sugar levels and to promote insulin secretion. There are several medications currently used to treat diabetes. Today, I want to introduce you to exenatide. It is a GLP-1 receptor agonist widely used to treat type 2 diabetes. Before we move on to our new targets, let's first look at how this GLP-1 receptor agonist works. This is a picture of the cell membrane of a pancreatic beta cell. And we notice that there is a GLP-1 receptor on the membrane. GLP-1 can act as an endogenous ligand and interact with GLP-1 receptor to exert its biological function, increasing insulin secretion. Scientists have discovered a drug called GLP-1 receptor agonists that can also interact with GLP-1 receptor and mimics the biological function of GLP-1. As a result, insulin secretion increases more effectively. This treatment method uses protein receptors as therapeutic targets. Today, I want to introduce a novel therapeutic strategy, targeting the insulin gene promoter region to regulate insulin expression. Now, let's look at how gene promoter region plays a crucial role in gene expression. Gene expression is a very complex process. Within this process, gene transcription serves as the first checkpoint, transcribing DNA into RNA, and the promoter is located upstream of the transcribed gene. Functioning like a switch that turns on the transcription of downstream genes. On the right side of this picture, we can see the transcription process more clearly. RNA polymerase II binds to the promoter region, also binding with transcription factors to initiate gene transcription. But what's important here is that upstream of the gene transcript, there is a special regulatory sequence called an enhancer. This special regulatory sequence has important biological functions that enhance gene expression. More excitingly, scientists have discovered a special enhancer sequence in the promoter regions upstream of the insulin gene that significantly enhances insulin gene expression. This sequence has been called insulin-linked polymorphic region, ILPR, and it holds promise as an innovative target for diabetes treatment. ILPR is also known for its unusual DNA structure, which is quite relevant to diabetes treatment. Now, please let me introduce the mysterious structure of ILPR to you. The DNA structure that we are familiar with is a double helix DNA structure discovered by Watson and Crick in 1953. Two DNA strands twist like ropes, connecting through base pairing. A with T, C with G. However, there is something special about the structure of ILPR sequences. Scientists have found that ILPR contains numerous consecutive C and G bases, which prompt DNA to form unusual quadruple helix structures. The first unusual DNA structure I want to introduce to you is called I-motif. And this structure is formed in DNA sequences rich in cytosine bases. In this picture, you can see this quadruple helix structure and also the special base pairing mode. This DNA strand connects through hemiprotonated pairing, that's through C with C plus connections. That is quite different from the base pairing mode, A with T, C with G, as we previously discussed. The second unusual DNA structure is called G quadruplex. It is composed of DNA sequences rich in guanine bases. In this case, 4 guanine bases form a G tetrad through hydrogen bonding, stacking like building blocks and stabilized by potassium ions. These two 4-stranded structures aren't just scientific curiosities. They do play key roles in regulating gene expression, and they are thought to be inspiring targets for diabetes treatment. Now let's look at how the ILPR structures is related to insulin expression. Scientists have found that the ALPR sequences in humans come in different variants. Here, I've listed a few of them. You can see that each repeat sequence looks just a little bit different from each other. And you might think, well, since they are all rich in guanine bases, they should all be able to form G quadruplex structures, right? But that's not the case. Out of all these variants, only 3 of them can form G quadruplex structures. And here is the interesting part. Scientists discovered that the most structurally stable one, called 1G, is linked to the highest promoter activity. In other words, it's the best at boosting insulin expression. In the previous induction, we learned that the expression of insulin is closely related to the stability of these mysterious DNA structures. This discovery offers a new approach to diabetes treatment. We can design small molecules to stabilize these 4-stranded structures, thereby activating promoter activity and promoting insulin gene transcription. That's like turning on the insulin production switch at its source, a completely novel therapeutic strategy. However, scientists do face some challenges when targeting these unusual DNA structures. The first challenge is the dynamic nature of nucleic acid structures. For example, the emotive is quite sensitive to pH change in the body, and that means even a slight change in pH can significantly affect how the ligand interacts with the DNA structure. This kind of instability makes it much harder to develop effective and reliable drugs. The second challenge is that designing ligands that specifically bind to these structures is difficult. And this isn't unique to this case. It's actually a common problem in many areas of drug discovery. Scientists must take many factors into account, which means that only a few number of molecules have the potential to become real drug candidates. Nevertheless, targeting the ILPR is still full of promise. With advances in science and technology, we have reason to believe that these remarkable 4-stranded DNA structures, like molecular origami, could lead to breakthrough process in the treatment of diabetes. Thank you for your listening. Today, we've explored a new solution for diabetes hidden in the fascinating world of DNA origami. If you have any questions or insights about this topic, please leave them in the comments section. I wish you have a wonderful day. Bye bye.